of the mature group.
Yeah.
So what's about to happen right now is really a reflection of the way things have been going at the BioBE Center. Last year we were invited to give a talk, and Charlie said, I really think that we should give a joint talk. And I said, okay, we can give talks back to back. Somebody goes, you know, like we just saw, somebody goes the first 15 minutes and somebody goes the second 15 minutes. He said, no, we should give a talk together. And I said, no way, there's no way I'm doing that. And this year he said, we should give a talk together because that's what architects do. And I said, well, this isn't a group of architects only, and this is biology and architecture and engineering. And he said, okay, we'll do it the biology way again. And so today, I want you all to have empathy for my first time giving a true joint talk. And Umberto told me at dinner that if your partner's in an architectural firm, this is a really common format for giving a talk.
I'm actually the mature part of the group. You want to introduce folks?
Yeah, sure. So this is Charlie Brown. I'm Jessica Green, and we wanted to introduce folks from the BioBE Center that are here today. Adam Altrister, Ashley Bateman, James Meadow, Gwen Murick, Dale Northcutt, and Jason Stinson. So if you guys could stand up just so people know who you are and where to find you.
And they can answer the questions that we can't.
All right, so I'm going to let you introduce the topic of the talk.
Okay. Inventing building microbes. I read a book called The Invention of Comfort. I thought the book was great, and I thought the word using inventing gave a certain flavor to comfort that was really important. I took books by John Crawford. I took his timeline, which was quite extensive, and lopped off everything but the last 100 years. And I just want to give you an example. Heating and cooling systems were, Crawford claims, that it is influenced as much or more by social status than their technical capability. And that interplay of technology and social continued to this day. Chronologically, the next thing in order was the heating, ventilating, air conditioning, and lighting systems were designed to be better and better and better. The engineers were doing a great job. What happened was that the architects then had complete freedom. They were no longer dependent on having to make small windows, condensed forms. So engineering and architecture went in separate directions. At about the same time as that, there started to be lab testing for comfort. And comfort got defined as more narrowly. And as a result, we have buildings right now where the envelope doesn't respond to the loads. An example of unshaded west glazing is a problem. The envelope usually would take care of that in traditional buildings like we were just looking at. So we've dug ourselves a little hole, or a big hole maybe. Buildings use 40% of our energy use. They use 70% of our electrical use. And it's a huge problem from a global warming standpoint. The approach one example of approach that we're taking to solving this problem, I'm in a box up here, Mark, put me in a box. I've never been in a box before in my life. I'm always on the other side.
Charlie's in a box, so we won't have feedback from his mic.
So we're doing one thing that the energy building community is doing is what's called a net zero building, which is defined here in the bottom of the slide. So the question is, is this the idea of inventing comfort, or the idea of building microbiomes, is that where we want to go? We, meaning all of us here that are working on this topic. Where do we want, what direction should we go? What should our buildings be like? This is a quote from Robert Venturi, wrote a book called Complexity and Contradiction in Architecture. It probably, if I had to pick a single book that changed architecture in the last few years, it changed modern architecture into postmodern architecture. Huge change. Architecture occurs at the meeting of the interior and exterior forces. Architecture is a wall. The inside and the outside becomes a spatial record of the resolution of these differences. It's a drama.
Can you guys hear Charlie in the back? Is his mic turned up enough? Yeah, all right.
We're going to talk about an isolated piece, the skin. The skin of the building. This is a building, by the way. But, you can see it's got different size holes in it. Big holes, little holes, multi-story. It's several layers of steel, stainless steel skin on it. So when we're talking about skins, we're not talking about the normal range of buildings necessarily that one sees. This is by Tom Meyer. Okay. Now, so that was all the warm-up for our stuff. This is a hospital study. Jess will explain to you, but this is the fun part, which is the telltales that we had on the window. We analyzed hospital rooms with mechanical supply and exhaust, and one with a mechanical exhaust and a window supply. This is the window part of the test. The yellow things are telltale, so we can tell whether the wind air is moving in and out of the window. Sometimes it comes in at the bottom and goes out at the top, or vice versa.
So a lot of folks have seen this data before, but the real motivation about the focus of what we did for our first study at the BioBE Center was because Charlie really felt like if you care about net zero energy buildings, then we really need to be asking questions about the skin of buildings, and in particular this very first question about the role of natural versus mechanical ventilation. And briefly, we found in this very first study that design choices at the skin make a difference. If you have rooms that are mechanically ventilated, which are in the blue, they're not a subset of air samples that are outdoor air samples, which are in the green, so these are bacteria, and window ventilated samples fall somewhere in between. And when we compared those gene sequences to databases of known sequences from the human microbiome, the oral microbiome, the skin microbiome, that's what mechanically ventilated rooms look like, and this is becoming a common theme here in our community, finding this kind of result, and outdoor air look like what you find in dirt and in plants. So then we went on to another analysis of really trying to understand the skin of a building at the Lillis Business Complex in the University of Oregon. So Charlie actually designed a lot of the ventilation systems in this building, and that's one of the reasons why he wanted to study it. And he designed classrooms to have what I understand, I may not be using the word exactly precisely, but a passive ventilation system where there are louvers on the side of the classroom that let air in overnight. And Charlie wanted to understand what the biological implications of this design was. And I wanted to bring up that we designed these samplers to look at airborne bacteria in these classrooms, and this has come up in a number of these talks, is that we only were able to have the resolution of sampling air every 8 hours. So we designed something that had a decibel rating that was below a projector, but given this theme that keeps coming up at this meeting of how we really need to change the resolution of our sampling, so I want to thank Noah Furrier and Norman Peace, and also Mark Hernandez kind of got the BioBE Center up and running with air sampling, and I know that Bill Nazaroff is thinking about ways to design a faster temporal resolution. But this data here from the classrooms is just looking at 8 hours, every 8 hours, and what you're looking at in the top graph is the diversity or composition of the most abundant taxa and how it varies in the outdoor air, because what we were really interested in is comparing the relationship between outdoors and indoors. And I know from talking to the Bruns Lab and other folks that this understanding this relationship between outdoors and indoors is seeming to become increasingly important. And what we found in the middle here is that the rooms that had the design that Charlie used, which was a night flush, when you had a shift in the outdoor microbial community, these night flush rooms closely tracked that, but rooms that didn't have a night flush remained relatively stagnant until we opened the vents on Saturday. And we've also been developing at the BioBE Center, collaborating with Autodesk to come up with visualization tools to help people that aren't in architecture or biology or engineering understand our results and to get excited about this work. So this is the Lillis Business Complex, and this is looking at the exact same data, but using this general audience framework. So what you're looking at is, this is the outdoor air, this is how the composition of the rooms with the no night flush change with time, and these are called petal diagrams or rose diagrams, where the shape of those diagrams show you something about the overall bacterial composition. And if you compare that to rooms that had the night flush, you'll see that over time they much more closely track the outdoor shifts in the outdoor air. So Rob Knight's group is also collaborating with Autodesk, and this was a software tool called BioBE that we're still working on now. So next, Charlie's going to explain a little bit about what we found in this building, looking at offices, again, focusing on design choices surrounding the skin, and this is looking at dust samples in the bacteria and dust.
And these, each one of the circles is an office. The blue ones were the ones that were naturally ventilated. Naturally ventilated meaning having an outside supply of the skin, but they were mechanically exhausted. These offices only had mechanical supply and exhaust. And as you can see, while there's some overlap, there's some distinct differences between the two.
So if you look at this, the biological data a little bit more closely and put it into a constrained ordination diagram, so the blue are the window ventilated offices and the red are the mechanically ventilated offices, you see that you have really distinct microbial communities in these two different room types. And particularly you see that you have a much higher proportion of Deinococcus in the mechanically ventilated rooms, and a much relative to the window ventilated rooms. And in the window ventilated rooms, you have a lot of methylobacteria microbes, and I think Jonathan Eisen has ideas about what this means. So we didn't really know,
others you do too.
Yeah, don't really know what the implications of this or why we're seeing this pattern, but we've been having lots of conversations with folks here about that.
This is a diagram that shows the separation between spaces in Lillis. These diagrams are a little confusing down here. This is really meant to be two spaces together. These are two spaces apart. And what you can see is there's a definite change in the microorganisms content as you go from spaces that are close together to spaces that are far away. Those long, thin shapes automatically have a lot more area than the same 9 squares in a more compact shape. So getting whatever the balance is that we're looking for between exposure to the outside edge, like with ventilation, will depend a lot on what kind of building that we build or design.
And something that Charlie has taught me is how form and organization has really changed in the last 50 years, and that buildings used to need to be thin and long, and now they're fat and tall. Thank you. So we're having a lot of discourse at the BioBE Center about form and organization and the choices that designers make and how that impacts the built environment microbiome. So again, I'm going to show you these results using these tools for a lay audience. And if you compare offices that are side by side to one another, they have really similar microbiomes. But if you take an office that's a much farther walking path, it's going to have a different microbiome.
This is, so we've talked about letting the air in at the skin. Well, if you let the air in, you let it out, you have to let it out or the building blows up like a balloon. So we, in this particular model, which is a classroom building primarily, we put lower vents in here that lets the cool air in, and we let that, when it gets heated up, it's exhausted out of the top. It's called a night ventilation of mass strategy, and when we do that, we can eliminate the mechanical cooling system. Next. It has a big impact on the way this building is designed, because we called it the lung because it exhaled the air from that was being drawn in from the outside. It also lit the center of it. So rather than having a circulation zone down the middle of the building, we had a breathing zone down the middle of the building.
So the last thing that we wanted to talk about is the new direction that we're going using this awesome comfort chamber that Charlie's lab has developed. And it's your lab's comfort chamber, so I should let you describe it.
Well, it's unique characteristics, relatively unique, is we can control the surface temperature of all the surfaces except the glass door. We can vary the air temperature, we can vary the relative humidity, we can vary the wind speed. So if you want to know whether people are going to be comfortable or not, the last group presented what it's like to be in a humid, hot place. We can simulate that. Okay.
And also, so every single one of these panels, you did say everything about your comfort chamber, and I know these things. So every single one of these panels can be put at a different temperature. So if you want to look at spatial environmental variation within this box, you can. And it's my understanding that this is really important in terms of understanding the potential implications of net zero energy, is understanding what the thermal comfort zone means. And if we decide as a society that we don't want to be bound within the thermal comfort zone that we're all used to now, this comfort chamber is one way of trying to think about if we can redefine what human comfort means. And we also want to know that what are the biological implications of varying environmental conditions inside. So we've started doing some preliminary experiments in this comfort chamber to first ask really basic questions. So here's Ashley, where we just divided the chamber in half, and we wanted to ask a simple question: can you measure a person's microbial aura? So if you reduce the biomass as much as possible, and where you have the same input air coming in on both sides, is it even detectable? Do we have, because we know it's kind of hard in this community to get enough biomass and air samples, for example, so we wanted to explore that. And so we had air samplers and Petri collection dishes, which we were inspired by the Berkeley group that from their research. And so what we did was we had in our preliminary experiment an unoccupied and occupied side of the chamber, where we again had these settling dishes around each occupant. We had particle counters, and then we had air filters at about mouth height if somebody's sitting down, and as close as we could get to the floor. And we were totally blown away that in just 4 hours you can see a really big difference between the occupied side of the chamber and not. And we had 3 people that sat in these chambers. They're all here in this room. I can't say who they are, but they're in this room. And if you look at these different people in our center, There's two people that have a really distinguishable microbiomes, their aura, so you can differentiate them from one another. And there's one person who's kind of all over the map. So we also have a messier but detectable pattern at 2 hours. And something that we're interested in is exploring how the human microbial aura, or the direct output of a person, varies if you change environmental conditions like relative humidity and temperature and airflow. And then we're also interested in the exchange between the built environment and humans, because we all know that's an important question.
Jessica and I, now, I don't know how many years since, it seems like it's been forever. But I wanted to explain our working relationship. And on the left-hand side, I have the capital A architect. A capital A architect is one that thinks he or she is quite good at the job. So this is a small A architect is someone more like me. The architect, you can tell, is on the left. He has his head in the clouds. He's got a fenestration pattern that looks like a bow tie. He's got rusticated high-heeled shoes, and he's firmly planted on the ground. This is my view of the microbiology professor. As a tower, it's a hot topic observation platform. This is a 360-degree insight tower. This is his dredge for data collection. He's got an all-terrain method. And of course, a truth anchor. So those are the players. This is the play. Listen and rejoice. The world is full of microorganisms, and we know little about them. How many microorganisms are there in a building? Oh, about 1 billion. I guess I'll have to add more closets.
So on that note, Charlie's always very good humored about working with us biologists. He's been very patient, and I think one of the it's been a journey, and I think one thing I wanted to share with this community that I'm learning is true architects aren't as interested in publishing papers. They want to make buildings. They want to design stuff. And so I think that that's something we need to consider in this community, is what are different types of metrics that are going to have impact on this field. And I think that Charlie says, well, I'm designing hospitals, so why don't you guys start designing with me? And then we can evaluate what we've made later. So it's a really different perspective, his perspective of doing stuff.
And so we would like to leave the group with the question, a little discussion about using the net zero model. What would a net zero microbiome building be? What characteristics, what performance? So we're open for questions.
And so I would say that in terms of what's important, I feel like it's important in this field. I feel like it's important not just to link what's going on in the built-in microbiome and human health, but also think about what's happening as our planet gets warmer and warmer and design choices are made to deal with that. What are our design choices going to mean in terms of the microbiome, and does that matter for our health? And we have time for questions. Yeah.
Question in the back?
Yeah.
Jessica, I wonder what were the three ovals in the unoccupied half of the comfort?
The 3 ovals in that. Should I pull that side up? And I've got to say that James Meadow is the guru. He's analyzed this data and can help conceive the study with others in the BioBE Center. So
I took it that the 3 ovals on the occupied side were the 3
oh yeah, I'm sorry if I wasn't clear about that. So the different colors On the occupied side are the different people, and the three ovals are different people. So there's two people that have a statistically significantly different.
So I get that.
Oh.
What's the unoccupied?
What are the ovals on the unoccupied side? So the ovals in this are essentially a confidence interval around the centroid of that. Right. So, the ovals in there, the way we did them here, are essentially a confidence interval around where you could expect to see these communities fall out. And so we did these in sequence, and we know that there's a big difference in temporal or in microbial communities over time. And so that's why we always had an unoccupied control. And so what this is essentially showing is that different times in the unoccupied side are more or less in the same field. There's a lot more overlap there, whereas two of the three people had very consistent microbial communities, even counting at the time. And a third person has maybe a less consistent signal, but it's very much predictive of the occupied side of the room. And these, by the way, is worth mentioning, these switch back and forth so that we can control for any sort of room side effects.
So I just had a simple question. I might have missed it. So this was just bacterial.
Yes,
I'm
sorry, yeah, this is bacterial.
And then
All of this is bacterial, and all of this is bacterial, and we really are plan on moving in the fungal direction, absolutely.
then I have a suggestion for nomenclature. You said microbial flora. And I would suggest microbial terroir. Because it fits the wine in your steak.
Okay.
It is a common term. We have a personal microbial terroir.
Yeah, like that.
If I could add to that in the indoor environment arena near Fukushima, we call it the personal cloud. So I'm calling your analysis of microbiome personal cloud.
Okay, all right, so. Maybe Eugene is really